Hello everyone, welcome to the module on the nervous system. In this module, we'll talk about the anatomy aspect of the nervous system, in which we will cover the different type of cells present in the nervous system. Okay, so starting with the first and the basic fundamental unit of nervous system, which is neuron. Okay, so neuron is a fundamental unit of the nervous system. Now, neurons are the signal transmitting cells of the nervous system, which are permanent cells. Okay, so it is a permanent cell. That means these do not divide in the adulthood. Am I clear? Now, the signal relaying cells with dendrites, which receives the input. That means these neurons have certain structures called as dendrites. Okay, this is the cyton, these are the dendrites. These dendrites receive inputs from the different neurons and it transfers through the cell body to the axon, which sends the output. Okay, so it receives input via dendrites and sends output via the axon. Am I clear? Now the cell bodies and the dendrites can be seen on the Nissl staining. Now, why can it be seen on Nissl staining? Because it contains rough endoplasmic reticulum. Okay, so rough endoplasmic reticulum stains during Nissl staining, and hence the cell bodies and the dendrites are seen by Nissl staining. Am I clear? Now, rough endoplasmic reticulum is not seen in the axon, and hence it does not stain by Nissl staining. Now, the neuron markers include neurofilament protein and synaptophysin. Okay, so these are the two important neuron markers. Now, talking about the other type of cell, that is astrocytes. Now, astrocyte is one of the most common glial cell type in the central nervous system. Now, it physically supports, repairs extracellular potassium buffer. Then it removes a lot of excessive neurotransmitter and is a component of the blood brain barrier. This also acts as a glycogen fuel reserve and acts as a buffer. Am I clear? So it is the most common type of glial cell type which has a variety of function. Now there can be reactive gliosis in response to neural injury. Okay, so whenever there is a neural injury, there can be reactive gliosis. Now, astrocytes are derived from neuroectoderm, and the astrocyte marker is GFAP, that means glial fibrillatory astrocyte protein. Is this clear? Now, talking about microglia and ependymal cells. Okay, so microglia cells are actually the macrophages or the scavenger phagocytic cells of the central nervous system. Now, these are activated in response to tissue damage, which release different inflammatory mediators, example the nitric oxide, glutamate, and these are not readily discernible by Nissl stain. That means these are not identified by Nissl stain. Am I clear? So these are basically phagocytic scavenger cells or the macrophages of CNS. Now, there can be HIV infected microglia which fuses to form multinucleated giant cells in the central nervous system, which is seen as HIV associated dementia. Okay, so remember that if there is a HIV infection which spreads to the CNS, there can be multinucleated giant cells in the CNS due to fusion of microglial cells. Am I clear? We're talking about ependymal cells. These are ciliated columnar epithelium or ciliated columnar glial cells which lines the inner side of the ventricles and the central canal of the spinal cord. Okay, so it lines the ventricles and the central canal of the spinal cord. The apical surfaces are covered with cilia, which circulates the cerebrospinal fluid, and microvilli, which help with CSF absorption. Okay, so the cilia helps in circulation of the CSF, whereas the microvilli help in CSF absorption. Now, there is a specific kind of ependymal cells or modified ependymal cells called as choroid plexus. Now, these are the cells which produce cerebrospinal fluid. So, remember the choroid plexus produces cerebrospinal fluid. Now, talking about the myelin sheath, okay, the myelin sheath is this structure, okay. Now, the myelin sheath increases the conduction velocity of the neuron down the axons. Now, how does it increase the conduction velocity? It increases the conduction velocity by saltatory conduction of action at the nodes of Ranvier, okay. So, due to the nodes of Ranvier, there is saltatory conduction, that means a jumping conducting movement from nodes of Ranvier, where there are high concentrations of sodium channels, and hence there is excessive conduction of the nerve impulse. Is this clear? Now, in the central nervous system, including the cranial nerve too, myelin is synthesized by a type of cell called as oligodendrocyte. Whereas in the peripheral nervous system, including cranial nerve 3 and cranial nerve 12, the myelin is synthesized by Schwann cells. I'll talk about Schwann cell in just a while. So, in central nervous system, it is derived from the oligodendrocyte, whereas in the peripheral nervous system, it is synthesized by Schwann cell. Am I clear? The myelin usually wraps and insulates the axon and hence increases the space constant and conduction velocity. Okay, so remember that it increases the space constant and increases the conduction velocity. Whereas the Schwann cells that produce the myelin sheath of the peripheral nervous system, these promote axonal degeneration. Okay, so these are the major aspects for axonal degeneration, and Schwann cells are the major injured parts of the nervous system in Guillain-Barré syndrome. Okay, the Schwann cells are derived from the neural crest cells. Now, what are neural crest cells? Please check the embryology module to know more about the neural crest cells. Is it clear? I'm talking about the last type, that is the oligodendrocytes, which produce the axons or the myelin sheath of the central nervous system neurons. Each oligodendrocyte can myelinate many axons, okay, whereas a Schwann cell can myelinate only one axon. Is this clear? So, these can myelinate approximately 30 neurons, whereas Schwann cells can myelinate only one neuron. Now, these are the predominant type of glial cells in the white matter. Okay, so these are the predominant type of cells in the white matter and are derived from neuroectoderm. Now, it appears like a fried egg histologically and is injured in multiple different CNS problems, that is multiple sclerosis, progressive multifocal leukoencephalopathy, that is the PML, and leukodystrophies. Okay, is this clear? So, it is injured in multiple sclerosis, PML, and leukodystrophies. So, this is all about the different types of cells present in the nervous system. Thank you for watching this video. If you enjoyed the video, please click on the like button and do subscribe to this channel. Let me know in the comment section below which topics do you want me to explain. Thank you.